My name's Cynthia.
And I'm Silvano.
And today we're going to talk about protein structures.
There are 4 subgroups for a protein structure: primary, secondary, tertiary, and quaternary.
Primary structure is a polypeptide chain of amino acids. This is pretty much what it looks like, and every circle is an amino acid. The primary structure is held together by peptide bonds, so that means that between each of these is a peptide bond. There are 20 different types of amino acids. We have polar, non-polar, and electrically charged. In a polar amino acid, you'll see mostly carbon and hydrogen. In a non-polar, there will be carbon and hydrogen primarily, but you'll see either oxygen, nitrogen, or sulfur somewhere in there. And in an electrically charged amino acid, you'll see an electric charge somewhere in there. Every single amino acid has this structure. We have a hydrogen up here, a base carbon, our amino group, our carboxyl group, and a side chain. And the side chain is what distinguishes every amino acid from each other. A chain of less than 50 amino acids is a peptide. A chain of more than 50 amino acids is a polypeptide or a protein. So just one thing I want to add for the electrically charged amino acids: if it's positively charged, it's a base, and if it's negatively charged, it's an acid. So an acidic amino acid or a basic amino acid. Yeah, okay.
The secondary structure consists of alpha helices and beta pleated sheets. Within the helix and the sheet are hydrogen bonds. In the alpha helix, there are intrastrand hydrogen bonds. In the beta sheet, there are interstrand hydrogen bonds. Intrastrand means in between the strand itself. Interstrand means in between one strand and the other.
The tertiary structure of a protein is the 3D structure of the protein. So I know I'm not the best artist, but this is pretty much what it looks like, except it's usually a little bit closer together. So as you can see, we have our alpha helices here and our beta pleated sheets. The interactive forces are between the side chains in the structures and between the structures themselves. So we have our disulfide bridge, and this is actually going from strongest to weakest. So we have this disulfide bridge, ionic bonds, hydrogen bonds, and van der Waals, and this is pretty much what keeps the structure close and together. The structure of a protein is not random. Although it may look random, it achieves maximum stability and lowest energy state.
The quaternary structure is made up of 2 or more subunits, these protein subunits. And these are all protein subunits. The interactions between protein subunits are disulfide bridges, salt bridges, and hydrogen bonds.
So now that we know what the final structure of a protein looks like, we can talk about the denaturization of a protein. So whenever you lose one of the first 3 structures, you denature a protein. Changes in pH, temperature, dehydration, etc. These can cause the denaturization of a protein. When you denature a protein, it loses its biological function. Thanks for watching.